prevalence and intensity of forest fires have become a pressing global issue, deeply tied to accelerating impacts of climate change. Over the past decade, the frequency and scale of these fires have surged dramatically, fueled by rising temperatures, prolonged droughts, and unpredictable weather patterns. Beyond the destruction of vegetation and wildlife habitats, forest fires produce pyrolyzed organic materials that linger in the environment, complicating the recovery process of affected ecosystems. The purpose of this project is to address this complex problem by harnessing the capabilities of pyrolyzed fungi and their unique enzymes to create sustainable bioremediation strategies. Forest fires are natural phenomena that have shaped ecosystems for millennia, but their recent escalations in both intensity and frequency has transformed them into significant ecological and societal challenges. Historical wildfires, such as the devastating 2019 and 2020 Australian fires and the 2010 Russian wildfires, exemplify the profound impacts of these events. They resulted in the destruction of millions of hectares of forest, the displacement of countless species, and the emissions of vast quantities of carbon dioxide and other greenhouse gases. These incidents underscore the delicate balance between natural system and anthropogenic influences. Sakiniz, yapacağız. While periodic forest fires can play a beneficial ecological role by promoting biodiversity and soil nitrogen cycling, the growing prevalence of under-controlled, high-intensity fires threatens the ability of ecosystems to regenerate naturally. The Mediterranean basin, for example, has faced a surge in several wildfires exacerbated by prolonged heat waves and human intervention that failed to account for biodiversity. Forests should be understood as increasing ecosystem rather than mere cluster of trees. Restoration efforts, therefore, must prioritize ecological dynamics to ensure long-term sustainability. A critical outcome of forest fire is the formation of pyrolyzed organic materials, which are carbon-rich substances created during combustion. These materials are remarkable persistence, often remaining in the soil for extended periods due to their complex polyaromatic hydrocarbon structure. While pyrolyzed organic materials contribute significantly to the soil carbon pool, it also poses challenges for microbial communities, soil health, and overall ecosystem recovery. The persistence of pyrolyzed organic materials are influenced by the conditions of its formation, including the temperature of pyrolysis and the chemical composition of the burnt material. At higher temperatures, the structural complexity of pyrolyzed organic material increases, making it even more resistant to degradation. This persistence creates the bottleneck for nitrogen cycling, potentially hindering the recovery of post-fire environments. Among the natural mechanisms that emerge in response to forest fires, pyrolyzed fungi stand out for their ability to thrive in fire-affected environments. These fire-loving fungi play an essential role in breaking down pyrolyzed organic materials, facilitating nitrogen cycling and shaping microbial succession. The genus Morchella, particularly the species Morchella exemia, is notable for its pyrolytic properties and its ability to produce carbohydrate-active enzymes.
Carbohydrate-active enzymes are a diverse group of enzymes that specialize in degrading complex polysaccharides, including those found in pyrolyzed organic materials. These enzymes, such as endo-1,4-beta-xylanase, endo-1,3-beta-glucanase, and endo-1,4-beta-mannose, target specific components of pyrolyzed biomass, enabling the breakdown of otherwise recalcitrant organic materials. This research focuses on leveraging these enzymes to accelerate the decomposition of pyrolyzed organic materials, thereby promoting soil health and ecosystem recovery.
To maximize the bioremediation potential of pyrolyzed fungi, this study employs recombinant to enhance the expression and activity of carbohydrate-active enzymes. By transferring the genes encoding these enzymes from Mortella eximia into the yeast strain, Saxarum cerevisiae, the project aims to produce larger quantities of these enzymes under controlled conditions. The choice of Saccharomyces cerevisiae as a host organism is strategies. As a well-established model organism in biotechnology, it offers several advantages, including its ability to perform post-translational modifications and its compatibility with a wide range of vectors. Additionally, its classification as a biosafety level 1 organism ensures safe and scalable production.
Third one is establishing a framework for ecosystem recovery. By integrating biotechnological tools with ecological principles, the project aims to develop practical strategies for post-fire soil remediation. The implications of this research extend beyond the immediate goal of pyrolyzed organic material degradation. Forest fires are expected to be more frequent and severe in the choice of cerevisiae as a host organism is strategic. As a well-established model organism in biotechnology, it offers several advantages, including its ability to perform post-translational modification and its compatibility with a wide range of vectors. Additionally, its classification as a biosafety level 1 organism ensures safe and scalable production. Objectives of the study. This project seeks to address several key objectives. First one is enhancing enzyme efficiency. By optimizing the expression of endo-1,4-beta-xylanase, endo-1,3-beta-glucanase, and endo-1,4-beta-monosidase, the study aims to improve their ability to degrade pyrolyzed organic materials. Second one is comparative analysis of enzymatic activity. The relative efficiency of these enzymes will be evaluated to identify the most effective candidates for pyrolyzed organic materials decomposition. Third one is establishing a framework for ecosystem recovery. By integrating biotechnological tools with ecological principles, the project aims to develop practical strategies for post-fire soil remediation. To maximize the bioremediation potential of pyrophyllous fungi, the study employs recombinant cloning to enhance the expression and activity of carbohydrate-active enzymes. By transferring the genes encoding these enzymes from M. eximia into the yeast strain Saccharomyces cerevisiae, the project aims to produce larger quantities of these enzymes under controlled conditions. The choice of cerevisiae as a host organism is strategic. As a well-established model organism in biotechnology, it offers several advantages, including its ability to perform post-translational modifications and objectives of the study. This project seeks to address several key objectives. First one is enhancing enzyme efficiency. By optimizing the expression of endo-1,4-beta-xylanase, endo-1,3-beta-glucanase, and endo-1,4-beta-monosidase, the study aims to improve their ability to degrade pyrolyzed organic materials.
The initial phase involved the collection of Mortella eximia, a pyrophilic fungal species, from a wildfire-affected forest shortly after the fire. The timing of this collection was crucial to ensure the acquisition of fungi actively thriving in post-fire environments. Once collected, the fungi were cultivated in nutrient-rich media, such as potato dextrose agar or malt extract agar, supplemented with specific carbon sources like xylan, mannan, or cellulose. These substrates serve to induce the production of the target enzymes endo-1,4-beta-xylanase, endo-1,3-beta-glucanase, and endo-1,4-beta-monosidase. Optimized growth conditions, including temperature, pH, and nutrient availability, were maintained to maximize enzyme yield. To isolate the genes encoding the target enzymes, RNA was extracted from Mortella eximia using the Trizol plus RNA purification kit. This process began by rapidly freezing fungal samples in liquid nitrogen to prevent RNA degradation. The frozen samples were then homogenized in Trizol region to dissociate proteins and nucleic acids, followed by the addition of chloroform to separate RNA from other cellular components. After centrifugation, the RNA-rich aqueous phase was carefully isolated and purified using ethanol washes and spin column filtration. The resulting RNA was assessed for quality and integrity using absorbance ratios and agarose gel electrophoresis. To obtain high-purity mRNA, the extracted RNA was further processed using the Thermo Fisher Dynabeads mRNA purification kit. The mRNA, selectively bound to magnetic beads coated with oligo-DT, was eluted after several washing steps, yielding a clean template for downstream applications. The isolated mRNA was reverse transcribed into complementary DNA using the high-capacity cDNA reverse transcription kit. Oligo-DT primers were employed to specifically target the poly-A phase of eukaryotic mRNA. The reaction conditions were carefully optimized to ensure efficient and accurate transcription, yielding cDNA suitable for polymerase chain reaction. Specific primers were designed for the amplification of the target genes encoding the enzymes. The primers incorporated recognition sequences for restriction enzymes to facilitate cloning into expression vectors. PCR amplification was performed under optimized conditions, including initial denaturation, annealing at gene-specific temperatures, and extension steps. The amplified products were analyzed using agarose gel electrophoresis to confirm the expected sizes. The amplified cDNA was cloned into the yeast expression vector PRS327, a multi-copy vector with the LYS2 marker gene. The PRS327 plasmid, selected for its high stability and compatibility with Saccharomyces cerevisiae, underwent indigestion with restriction enzymes to create cohesive ends. Specific enzyme combinations were used: Hindal and Notal for endo-1,3 beta-glucanase and endo-1,4 beta-mannosides, and Ecori and Sac-I for endo-1,4 beta-xylanase.
Ligation reactions were performed using T4 DNA ligase under optimized conditions. The recombinant plasmids were then transformed into Saccharomyces cerevisiae BY4742 strain using the Liag-SS carrier DNA PEG transformation method. This method enhanced the permeability of yeast cells, allowing the uptake of the plasmids. Successful transformants were identified by their ability to grow in lysine-deficient media, as the LIS2 marker gene complemented the azotropic deficiency of the host strain. To express the recombinant enzymes, transformed yeast cells were cultured in synthetic media supplemented with glucose, raffinose, glycerol, and ammonium sulfate. After incubation, the cells were harvested and lysed using glass beads, ensuring the release of intracellular proteins. Protein purification was achieved through immobilized metal affinity chromatography using a hystrap column. After washing in elution, further purification was conducted using gel filtration chromatography. Protein purity was confirmed through SDS page, which is essential for characterizing the expressed proteins. Enzyme activity was assessed using spectrophotometric assays with substrates specific to each enzyme: laminarin for endo-1,3 beta-glucanase, xylan for endo-1,4 beta-xylanase, and mannan for endo-1,4 beta-mannosidase. The reactions were conducted at 50 degrees centigrade, and the production of reducing sugars was monitored by measuring absorbance at 590 nanometers. This allowed quantification of enzymatic activity in units representing the amount of substrate hydrolyzed per unit of time. The results demonstrated that all three enzymes are critical for the degradation of pyrolyzed organic materials, emphasizing its role in post-fire soil remediation. Unit activity, defined as the amount of enzyme required to produce 1 micromole of product per minute, was calculated to compare the efficiency of the enzymes. The results provided insights into the relative effectiveness of each enzyme in degrading pyrolyzed organic materials. By combining traditional molecular biology techniques with innovative biotechnological approaches, the study developed a robust framework for exploring the potential of pyrophilous fungi in post-fire soil remediation. The detailed methodology ensures reproducibility and provides a foundation for future research aimed at optimizing enzyme activity and scalability for environmental applications. The increasing frequency and intensity of forest fires represent one of the most pressing challenges posed by climate change. While fire has historically played an essential role in maintaining ecosystem health and biodiversity, the scale of modern wildfires often exceeds the regenerative capacity of natural systems. These fires leave behind pyrolyzed organic materials, which, while rich in carbon, are notoriously resistant to decomposition. This resistance disrupts nutrient cycling, compromises soil health, and hinders the recovery of affected ecosystems. Addressing this issue demands innovative solutions, and this study offers a promising avenue by exploring the enzymatic potential of pyrophilous fungi.
The study focused on 3 carbohydrate-active enzymes. These enzymes target key structural components of pyrolyzed biomass: xylins, glucans, and mannans. Cloning the genes encoding these enzymes into Saccharomyces cerevisiae and optimizing their expression demonstrated scalable enzyme production and activity against pyrolyzed organic material. These enzymes degrade pyrolyzed organic material, making nutrients available for microbial communities, accelerating carbon reintegration into the soil, and supporting plant regrowth and biodiversity recovery. Beyond these specific enzymes, this research showcases the potential of recombinant DNA technology to enhance naturally occurring enzymes, paving the way for tailored bioremediation strategies. Future studies could explore enzyme cocktails, engineered variants for field stability, and CRISPR-Cas technologies to improve fungal genomes for pyrolyzed organic material degradation without disrupting ecosystems. This research offers insights into global post-fire management and ecosystem recovery strategies. As wildfires increase due to climate change, scalable, sustainable solutions are urgently needed. Traditional restoration approaches like reforestation are costly and limited in effectiveness. By enhancing microbial and fungal succession, biotechnology provides an eco-friendly, cost-effective alternative. In conclusion, this study advances solutions to challenges posed by wildfires. Harnessing the enzymatic potential of pyrophilous fungi offers a scalable, nature-based approach to accelerate ecosystem recovery. The research not only deepens our understanding of post-fire ecology, but also demonstrates biotechnology's transformative potential in addressing environmental issues. Future research could integrate bioremediation with carbon sequestration strategies. Since pyrolyzed organic material is a major carbon sink, its decomposition could aid greenhouse gas mitigation. Combining enzymes and microbial agents offer a promising approach to link ecological restoration with climate mitigation efforts. However, questions remain about long-term ecological impacts. Can recombinant enzymes be applied directly without disrupting microbial communities? How do fire intensity, soil composition, and climate affect enzymatic bioremediation? Addressing these questions will refine the applicability of these findings on a larger scale. In conclusion, this study advances solutions to challenges posed by wildfires. Harnessing the enzymatic potential of pyrophilous fungi offers a scalable, nature-based approach to accelerate ecosystem recovery. The research not only deepens our understanding of post-fire ecology, but also demonstrates biotechnology's transformative potential in addressing environmental issues. As climate change impacts intensify, innovative approaches like this will be vital for building resilience and sustaining ecosystem health. Collaborative efforts between researchers, policymakers, and conservationists can turn these insights into actionable strategies that support both ecological recovery and climate adaptation, offering hope for a balanced, resilient future where nature and technology work together.